Genetics and Genomics in Medicine. Chapter 5. Single Gene Disorders, Inheritance Patterns, Phenotypic Variability, and Allele Frequencies. Module 3. Uncertainty, Heterogeneity, and Variable Expression of Mendelian Phenotypes. In our previous module, we have discussed the different modes of inheritance for phenotypes that are determined principally by single genes. Some complications or considerations were discussed, including the effects of X inactivation in females, homozygosity in males, and occasional differences between homozygotes and heterozygotes for autosomal disorders. The occasional expression of disease symptoms in carriers of an autosomal disorder, the mimicking of autosomal inheritance by genes in the pseudoautosomal regions of the X and Y chromosomes, and the unique features of mitochondrial inheritance. In this module, we will discuss the broader implications that relate to uncertainty of mode of inheritance and difficulties posed by the heterogeneity in the links between DNA variation and phenotypes. In addition, we will consider how affected individuals within a single family can show variable phenotypes for Mendelian disorders, which by definition arise from variants in a single gene. Let us discuss briefly some compounding factors that can give rise to variable phenotypes in Mendelian disorders. The first is small pedigrees. It is often difficult to accurately assign the mode of inheritance for a particular disorder when the family tree is small. One approach geneticists use in dealing with this is to look at aggregates of individuals that have the same disorder, and then to calculate the overall proportion of affected children, and this is referred to as a segregation ratio. So to just look at a collection of families that have this disorder and try to determine the mode of inheritance that way. It is good to note, however, that in the age of rapid next generation sequencing, we now are able to sequence an individual's exome or a family's exome very rapidly, so it really does help us narrow that down even in the absence of a large pedigree. Another factor that can compound Mendelian inheritance patterns is the genotype-phenotype heterogeneity. So there are different factors that can give rise to a phenotype being caused by multiple variants at a single locus, or perhaps it may be several different loci that give rise to a given phenotype. And that brings us to the notion of locus heterogeneity. There actually can be multiple loci that can give rise to a particular disease. Retinitis pigmentosa is an example that comes to mind. There are several different genes, and variants in each of these genes can give rise to the same clinical diagnosis of retinitis pigmentosa. And lastly, we have allelic and phenotypic heterogeneity. It is very important to realize that many genes have multiple variants that are described, sometimes numbering into the hundreds, and each of these phenotypes may be distinctly different, and it's not necessarily well understood what drives that, but observations have been made. So there are a variety of situations that can give rise to variable expression in Mendelian inherited monogenic disorders. Let's consider genetic mosaicism. And genetic mosaicism really is the manifestation of new mutations that occur post-zygotically. So mutations can occur in the somatic cells, and there can be actually de novo pathogenic mutations that can arise at any time in post-zygotic life. And as a result, as is shown by this slide, an individual actually is mosaic. The different cells within the body have variable genotypes, and that can give rise to distinct populations of cells that have different mutational spectra. Now, certain post-zygotic mutations, which often occur early in development, may also result in germline mosaicism. And a person who has a substantial proportion of mutant germline cells, so that would be post-zygotically in their gonadal cells, will not show symptoms of the disorder, but they will produce gametes that contain a variable collection of wild type and mutant gametes. And so the individual, because these mutations are occurring in the gonads, it's not somatic, it's not going to affect the tissues of the body, but for the fact that this individual will be producing either sperm or egg that will have a collection of variants, both normal and mutant. The slide depicts locus heterogeneity, and as we said earlier, locus heterogeneity refers to the notion that different genes can give rise to the same phenotype at the end of the day. So here we have an individual, and this has to do with genes that are involved in inherited forms of deafness. And so we can see that we have DEAF1 and DEAF2, and the ones that are shown in red indicate that these are the mutants, the variants that cause deafness. And so this is a disorder that is actually a autosomal recessive form of deafness, and you can have parents that both themselves inherited two alleles. So these are compound heterozygotes. These individuals have mutants in both the DEF1 and DEF2 genes, which ultimately leads to them themselves being deaf, but they can give rise to healthy children because those genes are distributed amongst the offspring. This slide helps to exemplify the concept of allelic and phenotypic heterogeneity. So in this particular example, we're considering the very vast locus heterogeneity for the Barrett-Biedl syndrome. And the different percentages and the different genes, and they all occur on different chromosomes, all give rise to a particular syndrome, but this can come off of many different genes. So you can see here that there's a huge variety of different genes that could come off of that. Another example of allelic and phenotypic heterogeneity that doesn't arise from different genes, but rather different variants within a given gene would be the variants when we consider beta thalassemia. And beta thalassemia results from a deficiency of beta globin and can arise by any number of different mutations within the hemoglobin beta chain gene, the HBB gene. So we can have allelic and phenotypic heterogeneity then that arises from several different genes that can give rise to the same syndrome, as in the case of Barrett-Biedl syndrome. And we can also have several different variants within a given gene that gives rise to a particular disease syndrome, and beta thalassemia and the multiple mutations in the HBB gene would be an example of that. And here we have another example. This table shows the very wide heterogeneity of clinical phenotypes that can result from mutations in the lamin A gene, or the LMNA gene. So the LMNA gene is a gene that produces a protein known as lamin, and the variants that have been observed in this particular gene can give rise to different modifications in the protein that it encodes. So as a result, there may not be proper post-translational modifications. As a result, there may be a truncated protein that is produced, and there are a whole host of different diseases and different expression patterns that are observed, but these all harken back to this one particular gene, the LMNA gene. This slide depicts a pedigree for a non-penetrant allele. So what is penetrance? Penetrance of a single gene disorder is the probability that a person who has a mutant allele will express the disease phenotype. Now, in dominant disorders, that seems quite simple and straightforward. It would seem that individuals that are heterozygous for a dominant allele should show the disease and therefore have 100% penetrance. But actually, in reality, that's not the case. And different factors can give rise to what we refer to as non-penetrance. And this pedigree shows an individual that is displaying non-penetrance in an autosomal dominant disorder. So the individuals with the red asterisk are asymptomatic for the disease gene and their carriers. They've inherited a mutant allele, ultimately from the affected great-grandmother in generation 1. But none of them expresses the disease phenotype. In this example, the disorder is evident only in individuals who have inherited a mutant allele from their fathers. Individuals that just show a red asterisk as opposed to a filled-in symbol inherited the mutant allele from their mothers. As described later in this module, epigenetic mechanisms known as imprinting can result in this type of parent of origin effect on the type of clinical phenotype that is observed. So particularly in this example, even though the inheritance was autosomal dominant, the allele had to be inherited from the father. If it was inherited from the mother, then the trait was not expressed. Another factor that has been observed to relate to penetrance is the variable age at onset in late onset disorder. So when you have a genetic variability, it may not actually manifest itself symptomatically in the patient at an early age. It may take time, and it may only manifest itself in adults or even in senior citizen age. And the slow progression of disorders may be due to several factors. One factor may be, for example, that as a result of the genetic variation, a toxic product is produced, and that this product needs to accumulate over time in order to cause its particular effects. And one thought about this, one example of this might be a mutation in a tumor suppressor gene, that would take some time for loss of that tumor suppressor capability to actually result in cancer. And that's one reason why the increased rate of cancer goes up with age. This particular slide shows the relationship between age of onset and the probability of developing Huntington's disease. So Huntington's disease is a classic example of late onset single gene disorders. In this case, mutant alleles produce an abnormal protein that is harmful to cells and especially toxic to our neurons. The loss of neurons is gradually, but eventually results in devastating neurodegeneration, which is Huntington's disease. The onset of symptoms typically occurs in middle to late life, but juvenile forms are also known. age of onset curves for late onset disorders are often used in genetic counseling to calculate the chance that the asymptomatic person at risk, so if you knew you were a carrier for Huntington's disease, for example, and you wanted to know what was the risk for you to develop the disease, the age onset curve can help to calculate the chance that a person at risk will develop the disease. In Huntington's disease, an unaffected person who has an affected parent will have a 50% a priori risk that decreases with age. If one is still free of symptoms at age 60, for example, the chance of developing the disease falls to less than 20%. Expression within families. In our previous slide, we talked about the extreme example of variable phenotype in the notion of non-penetrance, and that we require a certain inheritance pattern for an individual who has an autosomal dominant disorder to display a. But that is the extreme, and there are a variety of different factors that can produce variable expression of phenotypes within families, and they are the same that result in non-penetrance. And these include non-genetic factors such as epigenetic regulation and environmental factors, as well as modifier genes, other genes that can attenuate the phenotype either in a more or less severe sort of way. And then, of course, there is imprinting, which we mentioned briefly earlier in this course, and another phenomenon known as anticipation. So we will discuss these in the subsequent slides. This slide shows the variable phenotypes that are observed in a family with tuberous sclerosis. So tuberous sclerosis is an autosomal recessive disorder caused by mutations in either the TSC1 or TSC2 gene. These genes actually make up two subunits of a tumor suppressor protein complex that works in a signaling pathway to regulate cell growth and proliferation. As can be seen by this slide, the disorder affects multiple body systems with characteristic tumor-like lesions that appear in the brain, skin, and other organs, and is often associated with seizures and learning difficulties. However, as it is evident in this family from the northeast of England, there can be considerable differences in the expressivity of the disorder. And so this particular pedigree is very exemplary of that. It should take note on how this is demarked. We have a special legend here that's showing what exactly clinically the different patients that are displaying the disorder are manifesting. Interesting pedigree. This slide describes the main explanations for phenotypic variation occurring in Mendelian disorders. So on the left in A, the first explanation is intrafamilial variation in phenotype. Unrelated individuals with the same Mendelian disorder may often have different mutations, as shown by the red symbols, at the disease gene locus, with different consequences for gene expression and disease. So, as I was saying, in A, in interfamilial variation, that means between different families, we can have different variants. And so we see different phenotypes. In this particular example, they're occurring at different locations, and presumably this will result in different modifications of the encoded protein that occur at different points in the protein. And even at the bottom here, we show some type of variant that occurred that affected splicing, as is exemplified by these diagonal lines that show that we have now an interrupted, or really, in this case, a total loss of function allele here. So this protein would be totally non-functional because it does not exist. B shows, however, another type of example, which is within the same family or intrafamilial variation of phenotype. And affected members of the single family would be expected to have the same mutation at the disease gene locus, but can nevertheless still show differences in phenotype because of genetic or non-genetic factors. In the former case, the individual may have different alleles at one or more modified loci, which could give rise to a different phenotype. So even though the particular disease gene in question, the variant would be the same amongst the family members, they might have variation in their modifier genes. And modifier genes make products that interact with the primary gene locus so as to modulate the phenotype. And different alleles of a modifier gene can have different effects. Another explanation can be for intrafamilial variation. Non-genetic factors can explain the variation. An example is epigenetic regulation, in which the disease allele can be silenced by an altered chromatin conformation, as shown by the green hatched box, or by variable exposure to environmental factors, as shown on the far right green circle, such as a specific virus or chemical exposure during development in utero. Parent of origin effect, or anticipation. Some disorders show consistent generational differences in phenotypes. Disorders such as Fragile X, mental retardation syndrome, myotonic dystrophy, and Huntington's disease are caused by unstable mutations that are often referred to as dynamic mutations, whose characteristics can change after they undergo DNA replication. As a result, the phenotype can vary between affected individuals and families, but in a directional sort of way. That means, for example, it can be expressed at an earlier age and become increasingly severe with each new generation of infected individuals. This phenomenon is known as anticipation, and the molecular basis of this will be covered in a later part of this course. This slide depicts an anticipation phenomenon as exemplified in 3-generation family that are affected with myotonic dystrophy. The degree of severity increases in each generation. So you can see we have the grandmother on the right, mom on the left, and baby in the middle. The grandmother is only slightly affected, but the mother has a characteristic narrow face and somewhat limited facial expression. The baby, however, is more severely affected and has facial features of children with neonatal onset myotonic dystrophy, including an open triangular mouth. The infant has more than 1,000 copies of the trinucleotide repeat, whereas the mother and grandmother each have about 100 repeats.